When you hear the name Lilly, you probably think about 4 core areas. The first one is obvious, right? Diabetes with insulin, GLP-1s, and oral antidiabetics. Second one, oncology, Alimta for lung cancer, and more recently, a unique sarcoma drug called Lartruvo, which has an outstanding overall survival benefit. The third one is a new area for Lilly, immunology, where we have a psoriasis agent which has an amazing over 40%, 100% clearance of psoriatic plaques than in moderate and severe disease. The fourth one is neurodegeneration, which has been, I think, overall, over 25 years, a very strong motivational area to be in because of the high unmet medical need, and it's a neurodegeneration area. I guess you heard earlier about Parkinson's disease, which is one of the diseases of the brain. Today I will focus on Alzheimer's disease, which is the most common form of dementia. And if we look at Alzheimer's disease, I will cover 3 key topics. The first one is actually to talk a little bit more about the disease and how it's treated today. Secondly, then, talk about what do we know about pathophysiology of this disease. How does it start? What is driving it? And does that give any clues then how we could treat it? And in the third part is actually then, what is the reality in the clinical environment? And how do you do clinical trials? How do you select the patients? How long do you treat them? And how do you judge if you have an improvement or not? So let me start with Alzheimer's disease. And it's really a disease with huge implications for not only the person that is affected, but for the whole family and society. And there are millions of these patients in the US, and unfortunately, with the aging population, it's likely to double in the relative near term. And there are huge costs then of caring for these patients, emotional and of course also financial. The thing that happens in Alzheimer's is probably something that we all recognize. Your memory gets somewhat worse. Where did I leave my wallet? Where are the car keys? And these are, common daily life experiences. But of course, Alzheimer is much more than that. But it's really related to what we call cognition, which is memory and some other aspects. And then it's about function then. How are you functioning in your daily life? Can you go shopping? Can you drive a car? Or can you, go to the toilet and handle your daily activities? And these are dilemmas, I think, as human beings, because you are very used to seeing your near and dear, and they probably look the same even when they have Alzheimer's disease, but in a way they disappear. Their mind is changing so much that they're not the same persons, and they may not even recognize you, even if you are their son or daughter or whatever. So that's, the real dilemma, and you need to care for them, and in the end, of course, they cannot manage anything more or less. So it's interesting if you look at Alzheimer's disease, and I'm personally involved in some of the National Institute of Health programs together with Francis Collins then, and the interesting thing is that for some reason cancer research in the US has at least 10-fold higher grants than Alzheimer's disease. So cancer has really been an area where there has been a lot of breaking science and huge interest, and also then funding from the government to improve the situation. Alzheimer's, is coming now. I think there's a recognition that there is a gap in the funding of basic research, and also then the problem is increasing so much that something needs to be done. And if you look at the evolution of treatments, and this is a slide illustrating actually then what happens with the most common ways of actually dying in the US over the recent years, is that because of advances in therapeutics, in cardiovascular disease, in cancer, including the one that you just heard about, there is less risk to die in those diseases, but the risk of dying in Alzheimer's has actually gone up. And it's really the opposite trend against the life odds for that disease. And like I said, you can measure the cost and emotional problems of being a caregiver, but it's very clear that the emotional stress of a caregiver for Alzheimer's disease is very, very high, much more than the physical stress. And also to the right here on this slide, the cost for the society is enormous. And the problem could even be so that a major proportion of Medicare costs, etc., in the future will have to be spent then on nursing home care for Alzheimer patients. So a key thing is that how can we prevent this happening? Right, so how do you diagnose Alzheimer's disease? And this is a slide, if we start to look at the left, and there is an upper yellow curve there, which shows in a way the normal aging memory loss, which is slight, but some people, have the luck to become 100 years old, and they have a very healthy brain and a phenomenal memory still. If you look at the Alzheimer then curve, yes, it starts relatively flat, and what happens in the brain I will come back to, but the pathology probably starts a decade before you feel any symptoms. So it may be a little bit like cardiovascular disease and cholesterol. You can go around with high levels of cholesterol, you get the slow arteriosclerotic disease, and in the end then you get thrombosis, myocardial infarction, stroke, and you die. In Alzheimer's pathology, there is something else happening. It's something which we call amyloid that is accumulating in the brain, which is a toxic substance, which then leads to degeneration of your nerves, and they form what we call tau tangles. And if you look at the Alzheimer curve, then the yellow goes into blue. And the interesting thing with the human brain is that many of us have tremendous overcapacity of neurons. So you can lose a lot of neurons. In fact, for Parkinson's disease, some have said that you can lose 80% of your neurons, but you don't have any symptoms. And then, of course, it's a rapid decline then. And it's the same with Alzheimer's disease. The brain is compensating for a very long time for neuronal loss, but when you reach a certain threshold, you start to deteriorate. And in the end, then, you end up in the red line, where you more or less lose all of your capabilities. The diagnosis of Alzheimer's is a clinical one of dementia, and it's usually called Alzheimer's because it's the most common form of dementia. But there are several other types of dementias, and I think to be really puristic, the Alzheimer diagnosis is actually a diagnosis after the patient had died, and you do an autopsy and you section the brain and look how the brain then is behaving in relation to presence of amyloid plaques and these tau tangles. That's the final diagnosis. Today we can do better. We can actually see this in the living human brain using then brain imaging technologies. So, what do we think is causing this disease? And you know that there is a current revolution in our understanding of human genetics, and there has been particularly interesting studies done in Alzheimer patients. And most of these studies points actually to then that if you increase your amyloid load in the brain, your risk of getting Alzheimer's is much, much higher. In fact, there is what is called a Swedish mutation, which I think is particularly relevant to a Swede, or maybe an immigrant to the Midwest in the US, with Swedish genetics. But anyhow, this mutation is causing amyloid to accumulate at an early age. And the people with this get Alzheimer's disease when aged 30 to 40. It's this early onset Alzheimer's disease, which also, is seen all over the world in some rare families. The other opposite is also true, that if you have a mutation, so you have less amyloid formation in your brain from birth, then your risk of getting Alzheimer's is very much reduced. And there was a study recently done in Iceland on people that are 100 years old and have perfect brain health. And they have a mutation, so they have very little amyloid in their brain. And that's the basis, in a way, then, can we use this knowledge then to design pharmaceuticals to prevent Alzheimer happening or to treat the symptoms if you already have Alzheimer's? There has been several trials in that space using antibodies, and there have been some signs of improvement then of the decline of the disease, but I think so far these antibodies have not solved the problem, but they, I think, have given us hints that it may be in the right direction. So what does Lilly want to do in this space? We have a target in that before 2025 there should have been a real impact then on Alzheimer's disease. But what we also know is that we are trying then to be rational in this space, to use the genetic knowledge to then impact these disease pathways, preferably in a preventative way. But we also realize that even if you reduce progression of disease, these patients already have symptoms. And these symptoms, can vary a lot, but they include memory losses, behavior problems, and so on. And there is also a need to design new pharmaceuticals to affect those types of symptoms. So here, again, it's a summary of our strategy then, showing the two hallmarks. To the left, it's the amyloid plaques, and how can we influence formation of them, or even remove them, which could be a potential approach. It's the tau tangles, which are then the signs of the degenerating nerves in the brain. And we also then, like I said, are trying to find out new symptomatics that could help then, for instance, your memory. Today, Alzheimer's patients are treated with so-called acetylcholinesterase inhibitors, which increase the level of acetylcholine, a neurotransmitter in the brain, and has some enhancement of memory on a temporary basis, but as the disease is progressing, this benefit disappears. So you need something that is better than that. Here is a cartoon of various approaches that we are undertaking. And the first one is actually trying then using oral agents to prevent formation of the amyloid beta, which is the toxic peptide and a precursor then to the plaque formation. We are also doing things to neutralize amyloid beta and the plaque that is already there. And here you can take two different approaches. We have an antibody called sulanesimab, which binds free amyloid beta in the brain. And there is another antibody, which we have a code for N3pG, which binds to the plaque, triggers a microglia inflammation, which actually chews up and depletes, take away the plaques from the brain. There is also a link in some way, although not yet quite understood, between the amyloid plaque and the killing of the neurons with the tau then accumulation. And we also have in this space new agents that directly affect tau. And it's really an amazing story emerging in neurodegenerative diseases that even go back, to the prion story, that certain misfolded proteins can enter the brain and spread throughout the brain and be toxic for neurons. I guess you all remember the mad cow disease, perhaps, and the problems that were in England with people eating meat that was forbidden then for several years. And the meat coming from the cows actually then was contaminated by prions. And probably Alzheimer's may have some similarities to that. And I will briefly mention why. Okay, so how can we get some evidence that the new therapeutic approach could be beneficial in patients? And to do that, we use models in mice, often then mice that has been genetically changed so that they get amyloidosis, an accumulation of amyloid in the brain. So this is a mouse that has this Swedish mutation, and this means that when this mouse is very old, this mouse brain is full of amyloid. And to the left then, it's a section of the brain, and amyloid is stained with a brown stain. And you see it's completely full. And when you treat these animals with an oral BACE inhibitor and the antibody N3pG, you can clear out these amyloid from their brains. So this is something then that is a fundamental, I think, discovery that now will be tested in humans to see, can you remove this in a safe way? What also another question is that, yes, if you lose your neurons, are there any signs that you could kind of help them recover if they are damaged, or even come back, so-called neural restoration, so you can get back your memory? And in these animals, there is something happening that you see damaged neurons, which we call then dystrophic neurites, which are the brown swollen things there. And here then is a summary of what happens with these swollen neurites if you pre-treat with the amyloid, anti-amyloid agents. And either a BACE inhibitor or N3pG, or even better, the combination of them reduces then the signs of neuronal damage in the brain as well, which should be good if you could achieve that also in humans. So that was the amyloid theory. The tau, the neurofibrillary tangles, is something different, but like I say, is likely to be linked to the amyloid. And tau proteins are associated with microtubules, which is really a catering system, if I may say that, from the neuronal cell body along the axon with organelles and some other specific then functions to the nerve terminals. And if tau is hyperphosphorylated, it changes properties and it aggregates and it forms filaments and tangles in the end. And this is a misfolded protein. And coming back to my comments about prion and mad cow disease, these tau tangles have the property of spreading throughout the brain in some way. And we are trying now to stop this spreading by various then approaches. So here is a neuron which then has tau within it, and it's coming out of the neuron, and by having an antibody then being present there, we hope to bind the tau that is spreading and neutralize then the damaging effect. And here is just an illustration of an experiment then in rodents, where the blue bar is the control, and after various antibodies with code names, we have chosen the green one, which has the best inhibition, and this is now tested in humans in Alzheimer's disease to see then if we can prevent the spread of tau in the brain and potentially the symptom decline. So, how do we do then clinical trials in various ways? Well, first of all, Alzheimer clinical trials are categorized by being very long, very large, and have a very high failure rate. My dear friend and previous boss, Lechleiter, showed me actually from my window at my office and pointed at Lucas Oil Stadium. And he says, every time we do an Alzheimer trial, it's the same cost as this stadium. So it's really a huge bet, that you're doing when you're doing them phase 3 trials in this disease. But it's also, of course, a huge reward and a huge need that you could fulfill. So how can we do this better? And Lilly has taken a lead in the pharmaceutical industry in the world by having unique imaging agents that you can use then in the living patient to diagnose Alzheimer's. It's the amyloid detection that we have, and it's also the tau. And these agents can be used to select patients that actually have Alzheimer's disease. That's what we are interested to treat, and also has the pathophysiology that we are trying to influence. And we are excluding patients that don't have amyloid because they are unlikely to respond. And we also know that they can be very stable and they don't deteriorate in the same way. The tau imaging agent is somewhat different because this agent is actually highly interesting, and the signal in the brain is correlating to the symptoms. So if you have tau accumulation in a portion of your brain, it's also a correlation to the functional area that this brain represents. So, and I will show us a slide of this. So this, I think, is right now revolutionizing, in a way, also our understanding of brain function. And clearly, the Alzheimer's space is now looking very much into these agents then as helpful tools then, both in the diagnosis, but also how we do the clinical trials. So tau imaging in the brain is then a very recent, actually, addition to our opportunities to look at this disease. And in the normal person, there is some signal, but if you don't have amyloid, it's very little. And the interesting thing is that if you start to accumulate amyloid in your brain, you will have an increasing signal, particularly in the temporal lobe areas then which have cognition as the function. And here you can see then it's starting to get greener and yellow, which is a signal. And if you have fulminant Alzheimer's, it gets even red. And this color coding then is actually to help us to see increasing intensity of labeling. And that is something then that can be used in various ways. Today, we have a very laborious testing that we do with these patients. They go through a variety of mental tests and questionnaires. You also, of course, ask the caregivers and so on. But this could be an objective way of actually looking. Do you have a treatment here that can prevent this reddening happening? And are there regions in the brain that are protected more than others, etc.? Okay, so if we look then at how we do the Alzheimer trials, we can also think about, so when do these changes happen during the course of the disease? Like I said, Alzheimer's disease is probably a disease that you have without knowing it for a decade. Maybe your friends knows it, that your memory is not good. You forgot your wallet for the 5th time somewhere and you don't know where it is. But it's really something that can also be correlated then to when does the amyloid come and when does the tau come. Amyloid seems to appear then probably 10 years before neurodegeneration. And it's really something that can be monitored, like I said now. And what we have done is to use then these type of agents to study different parts of the disease continuum. And we usually call it then preclinical stage, when you have amyloid in your brain, but you have very little symptoms. Then you go into mild cognitive impairment or prodromal Alzheimer's, and then it's mild and moderate and severe disease. This is, I think, relatively helpful then to when you do clinical trials, but it's really a varying course also in different patients to remember. So what happens here then when we score our patients, and what can we look for? Solanezumab is an antibody then. It's one of these stadiums that equivalents that we tested. We have done actually 3 trials with solanezumab, and the dilemma was here that we felt there was a signal in 2 of the trials, but when we did the 3rd trial, the effect was too small. And here then is actually an illustration of solanezumab in red versus then placebo in blue. And there is a slight difference here, but it's not really big enough to be relevant. And why did this happen? And there are a number of reasons here that I have listed. And I think you could say first that, is this agent good enough to inhibit the amyloid? The unfortunate thing, when you look at brain treatments is that we have a blood-brain barrier, meaning that small molecules that are lipophilic and, agents like nicotine and alcohol go very rapidly into the brain. But proteins like antibodies, they only penetrate 0.1% of what you have in your blood. So there's very little that actually comes in. So that could be one of the reasons here. It wasn't really potent enough. Another reason can be that, yes, you had an agent, but the disease stage was too late. These patients that were studied here were so-called mild Alzheimer's, and I think we know that the plaques in the brain are more or less already maximal. So why do you think you will have an effect if the disease is already maxed out, kind of? Of course, you can say that the whole amyloid hypothesis may be wrong, and why wouldn't you try a tau agent instead? This is something that we actually are doing. The thing that you can show, though, is that instead of inhibiting the soluble form of amyloid with solanezumab, if you use a plaque depleting antibody like N3pG, you can see the following images then. And this is an Alzheimer patient where you see the brain from the side, and you see this staining of yellow and red, which is the higher density then of amyloid. And this is before the antibody N3pG. And if you look at the lower one, you see most of the signal has disappeared. So this, I think, is one of the first examples showing that you can remove the toxic amyloid from the brains of Alzheimer patients. And this is something that also Biogen has seen with their antibody. And the key question now, I think, is this safe? Can you take away the amyloid using actually then microglia inflammation as one of the mediators without having brain edema or other damage? And that, I think, is still a dilemma. So the second thing is, so if you take away the amyloid, will you get your memory and other behavioral capabilities back? And that still, remains to be seen. But I think it's highly interesting. It's another approach how you can address this. The third approach is to have an oral agent which goes then more or less 100% into the brain. And here is a so-called beta secretase inhibitor. And this agent reduces amyloid in cerebrospinal fluid, which is a marker, by more than 75%. And currently there are ongoing trials then with this agent in earlier Alzheimer patients to see if you prevent formation in a very effective way, will you then see some clinical benefits? And the Lilly trial then will read out in 2019. But there was somewhat discouraging a recent announcement from Merck, who has a similar oral beta secretase inhibitor in phase 3 trials. And they, during our trials, we have independent data monitoring committees that looks at the data and kind of advises then, do they see safety signals so we should change or stop the trial? Or they can also do what is called futility, meaning that if they look at the data and see that there is no chance of a positive trial, they advise us to stop. The Merck Data Monitoring Committee advised Merck to stop this trial because there seemed to be no chance of actually having a successful results. And these were in mild and moderate patients, which means it's relatively late in the disease history. At the same time, Merck is doing a trial in earlier patients in prodromal, and that trial is continuing, which to me suggests that maybe they saw something in this earlier part of the disease that makes it worthwhile to continue in this earlier stage. So we are very keen then and waiting on publicized data so we can understand, did they see a signal in the mild group? Remember, this trial was a combined trial in mild and moderate. And if the moderate doesn't work, the whole trial is likely to be negative. So we want to see this. So let me end with some comments then, and how can we be better and hopefully then have a preventative therapy that changes the disease progression in Alzheimer's. And there are several ways of thinking about it. Clearly, to use more powerful agents like I have described. Personally, I think an oral BACE inhibitor is much more likely to influence amyloid in the brain than an antibody that goes in 0.1%. So that's the first thing. The next is, can we use combinations of agents then to have a more effective response? The next is also using our imaging agents, which can help us to select patients that are more likely to respond. And also, when we have these images, can we see actually that something is changing in the brain of these patients? Are we seeing a reduction in amyloid, or are we seeing that the tau signal is stopping or even disappearing? So that is something that we are now looking for. And finally, I think we also need to modify in some way how we measure the cognition and function in these patients to make it easier, I think, for everyone involved in these trials. So this is the fourth core area of Lilly, as I said in the beginning. We have three other areas, perhaps fortunately, that can actually help us fund this fourth area. We are still very committed to this space based on the huge unmet medical need, and also, you know, I think the knowledge that Lilly has built up over decades is really a competitive edge for us versus other companies in the industry. So we really feel, you know, that this is one of the most difficult tasks, I think, in human research, to understand the function of the brain in health and disease. And it's also something that we all will be exposed for when we get older. And I think we all want to have a healthy old life with a great brain function. And I cannot emphasize enough, you know, that the patients are waiting, the caregivers are waiting. Clearly also, I think, payers, in a sense, the societal cost need something to then reduce the burden of this also financially. So with this, thank you very much for your attention.